So good afternoon. We're going to get started. It's a great pleasure to have Joan with us. He's from Barcelona. He's from Barcelona. He did his PhD in Barcelona, and then he completed a 6-year postdoc with Michael Karin. Most of you know he's one of the key leaders in modeling the link between inflammation and cancer using liver as the main working model. He's been extremely successful, was very productive with Michael Karin, always high in journals, and then he moved to the Fox Chase Center, which is based in Philadelphia, one of the NCA comprehensive cancer centers in the US, and he's been working on modeling or trying to integrate data from animal models in HCC with human samples and how to improve modeling development and work with animals and how we can use that in practice. So it's great to have you here. I'm looking forward to the talk.
So good afternoon. First of all, I would like to thank Augusto for the invitation and your kind introduction. And secondly, I would like to say that for me it's very special to be here today. So I've been working with Liver for 2 years now, and I recognize and appreciate Most as a very important leader in the field. And for me, probably you are one of the most knowledgeable audiences that I have. So for me, it's a special occasion. So today I'll be talking about how we've been trying to navigate liver cancer molecular complexities using mouse models. And I divided the talk in 2 parts. The first part will be longer. It's about how studying the cell of origin of HCC led me to be more concerned and interested in the differences and similarities between human and mouse HCC using genomics. And the second part is about an ongoing project we have in the lab about the role of SOX9 in cholangiocarcinoma development. This will be much shorter, so don't get scared if you go into it. So my first contact with the liver was, as you mentioned, when I was transitioning from my PhD, when I was studying epigenetics using Drosophila to a completely different area in Michael Karin's lab, studying liver cancer using mouse models. So the first project I was involved in was in the isolation of these hepatocellular carcinoma progenitor cells. These cells, we isolated these cells based on their aggregating properties from livers that they don't contain tumors, HCC, still, they only contain the preanaplastic lesions. But the observation that struck me the most to me was the fact that we found when we compared the gene expression profiles of HCPCs, they have a considerable similarity with oval cells. Moreover, oval cells also have the same property, aggregating properties. So you can isolate the oval cells based on their aggregation. So for me, this was an indication that might be that hepatocytes are not the origin of HCPCs, and therefore HCC, that maybe oval cells could be the cell of origin. This had been proposed for a long time by different people, but I thought that that was the right moment that maybe we could try to really answer these long-standing questions. So to do that, I decided to use the SOX9 CREATI2 for lineage tracing, because theoretically, with this tool, SOX9, you could label in a normal condition bile duct cells and potential resident stem cells. And during injury conditions, the same population of cells plus these oval cells or progenitor cells that are proliferating. So the hope was that with this tool, we could answer this question. The liver progenitor cells were the origin of cases. Unexpectedly, using this tool, I stumbled upon the hybrid hepatocytes. So I isolated and identified this new subpopulation of hepatocytes that are characterized by being periportal, and they express markers, vector markers in low levels, like SOX9, osteopontin, and others. So we characterize the behavior of this population in different conditions, homeostasis, and different mouse models of liver injury and regeneration. I do not go into the details. Also, in a transplantation model, we could see or demonstrate that hybrid hepatocytes are the population with the higher vegetative potential after transplantation. But going back to the original question, now we had a complication here because the SOX9 is not only labeling ductal oval cells, we have another population with hepatocytes that also label at the same time using this tool. However, we did experiments of lineage stretching in mouse models of HCC. We always try to inject the tamoxifen previous to any carcinogen treatment or before the onset of liver damage. So, for example, in the DN model, we inject the tamoxifen at P7, P14, the injection, and then analyze the tumors after 9 months. MAPUPA plus high-fat diet is a model that Hayato and Michael's lab developed while I was there. So we injected tamoxifen at P7. MAPUPA expression starts around 5 weeks, so the chronic liver damage starts by a month and a half. Then we fed the mice with high-fat diet, and at 9 months we analyzed the tumors. In the STEM model, we injected the streptozocin P2. This is supposed to kill specifically beta cells in the pancreas, so these mice become diabetic. Tamoxifen injection at P14, high-fat diet P24 months, very fast. All animals, male animals, develop HCC. So we did the experiment, and the results were very clear. We didn't find any single tumor in any model that was labeled with the reporter gene, the YFP, meaning that, closing the circle, that HCC does not originate from hybrid hepatocytes or ductal or oval cells. So that was the conclusion. But the problem I had trying to answer this question was that whether liver progenitor cells have the HCC, the problem I had is how we can generalize these findings. Because anybody can ask in which model. Right? We analyzed these in 3 models that we have in the lab, but how can we generalize these findings to HCC? And this was related to a problem that I was starting to recognize that in the field, we have an endless list of models available of hepatocellular carcinoma. We have models based on carcinogens, like the DN that we use, but we have many others, carbon chloride, aflatoxin. Then we have combination with diets. We have all the CD choline deficient diets. Then the genetically modified mice, knockouts, MDR2 is a classic, P10, and so on. Transgenic mice, even the sleeping beauty system, you can generate as many models as you like. Models that try to recapitulate the chronic infection of HCV or HBV, expression of HBx protein, and so on and so on. And moreover, we have an additional problem that when you look at publications using these models, it's very difficult to find 2 independent papers using the same model that model is exactly the same. So we have variations of dose, number of injections, different time points, the age of the mice can be different, genetic background, diet composition, and more. So it's like the snowflakes, it's impossible to find 2 identical snowflakes. So I think this is a problem we have in the field. And the reason for that is multiple, but mainly by the disease itself. As you know, HCC is 2 diseases in 1. There are different risk factors associated with the development of HCC. Not only that, there is a highly heterogeneous the incidence. So what can be important, like HBV factor, can be important in China, and the US is not so important, and vice versa. So the interest can be different depending on the risk factor. Also, these risk factors are changing over time. It's the case of BMI or obesity. Decades ago, this was not a big deal, and nowadays is a risk factor that is growing incidence and becomes more important. So new mouse models are needed. People is trying to generate these models, and we still don't agree which one is better than which. And finally, there is no clear oncogenic dominance in HCC, meaning that, for example, if we compare with pancreatic cancer, most patients have activation of KRAS and P53 activation, and then this led to the development of the KPC model. It has been very successful. It's very similar histologically, molecularly, all the stages of the disease. huge success, but we don't have that in HCC. HCC is much more complicated. I think this is one of the reasons we don't have right mouse models. So in our case, these were our choices. So we have the DN high-fat diet, TAG1, STEM, and MAP. These are the models we were using in Michael Karin's lab. But why? Well, I don't know very well. Usually it's historical reasons, but we don't have any evidence that which model is better than the other. So this is what it is. And any other lab can have a different set of mouse models. So to me, this was worrying because maybe we had some mind-side bias here. We tend to think that our models are better because it's the thing that we know, and we don't see the problem. So this led me to ask the question is how molecular similar are these mouse models to human cases? And at that time, all these papers using whole exome sequencing, whole genome sequencing of HCC started appearing in the literature. So I thought, why we don't do the same with our mouse models and we see if how they compare to each other, how they compare to human, and how can we use this information to improve the mouse model. So for this study, we decided to use the mouse models we had in the lab. So the DN that doesn't have chronic damage, no cirrhosis, time to cancer is 9 months, and histology is not very similar to human HCC. The MAP-UPA has chronic damage, cirrhosis is mild. In this case, I would say fibrosis to be correct. So mild fibrosis, and time to cancer is 9 months as well. Histology, 80% of the tumors are adenoma-like, only 20% are fully HCC. The STAR model, we have the chronic damage, severe fibrosis. Time to cancer is really fast, 5 months, and the histology is really human-like, malignant phenotype and really similar. And the TAG1, that is a deletion, albumin-3 TAG1, we have chronic damage, there is clear fibrosis, 9 months, and the histology mostly is adenoma-like as well, low malignancy. So we isolated 19 tumors from DN and STEM, and 9 tumors from the MAP and the TAG1 for a total of 56. We performed exome sequencing and RNA-seq. And then from the ICCC TCGA, we got exome, whole genome, and RNA data sets with the idea to compare mutational signatures, frequently mutated genes, expression profiles, and using this information to see the mutated pathways. So the first thing I want to show you is the mutational load. So we have 4 independent human cohorts, China, US, France, and Japan. Most of them have a similar mutational load, with the exception of Japan, that have significant higher mutational load. And if we look to the mouse models, we see a huge variability. MAP-UPA have a low mutational load, TAG1 similar to human, and then the N is clearly much higher. These are totally expected because DN is a carcinogen, is a mutagen, so we were expecting to have a lot more mutations. The surprise came with the STEM, because this model was supposed to work in the way that these mice become diabetic, the high-fat diet generates chronic liver disease, and this leads to the HCC development, but this was suggesting that the streptozotocin basically is mutating the liver very efficiently, because the mutation load is really crazy. So these are the mutational spectra for every case. We can see that humans tend to have very similar mutational spectra, with the exception of the Chinese cohort that have an enrichment in T2A mutation type. TAG1 and MAP are models that have very similar mutational spectra compared to the human. DN have an enrichment in T2A. This was expected because the mutation process is already known for DN, so we are expecting to see these type of mutations. And again, STAM clearly a huge enrichment on C2T, meaning that this is clearly a mutagen, is basically mutating the liver very efficiently. So next, we try to understand these mutational spectra that I just showed you, where was more likely to happen? So which mutational processes were generating these mutational spectra? So this analysis, the idea behind is that the final outcome is coming from the composition or the interaction of different mutational processes, different mutational signatures, like aging or any mutagen or whatever you want. These mutational signatures are well characterized, so you can try to figure out this final outcome, what's the most likely combination of signatures that can lead to this function. So these are the already characterized signatures. And here we have the different human cohorts and the mouse models. We are trying to cluster them based on the mutational signatures present in their mutational spectrum. First thing I want to highlight here is that the MAP-UPN tag1 models cluster together with the human cohorts, meaning that the processes that lead to the mutation in these models are very similar to the human. The DN models have basically only one signature that is itself, so it's the same signature of the mutation that is known. And then the STEM model is clearly enriched in signature 11 that is alkylating antineoplastic agent. Streptozocin is an alkylating agent, so this clearly was demonstrating that streptozocin is clearly mutating the liver. And that was the mechanism. So then we try to map the mutations in the mouse models in the genes. So here, every column is a mouse tumor sample. Upper panel, we have the most frequently mutated genes in human. And here in the lower panel, we have the most frequently mutated genes in mouse that are not commonly mutated in human. This is dividing the mouse models, and you can see clearly that tag1 and MAP, they don't have very few mutations. We don't find any single mutation in these mouse models with the typical genes found in human HCC. If we look to the genes most commonly mutated, p53 beta-catenin, p53, the STAM is the only model, but just 2 samples, so it looks like it's difficult to find p53 affected in mouse models. Beta-catenin, STAM is the only one, and here we have several cases in which we have the typical activating mutations of beta-catenin. That was really encouraging, as well as, for example, AXIN1, 2 nonsense mutations are AXIN1. And the last thing I want to highlight from this figure is that the DN model is completely dominated by BRAF V600E mutations. The problem is that BRAF V600E is never found in human HCC, although in this model it's clearly dominating this one. Now, if we map these mutations in signaling pathways altered in HCC, as expected, so here we have the gene presented in human and the models. The MAP tag1, obviously there were no mutations, everything are zeros. And the DN and STAM, they have a lot of mutations, so we have enrichment, especially for the STAM, in the typical pathways like Wnt pathway or cell cycle and chromatin fire and so on. So, but this is somehow expected because so many mutations that it's easy that you will map them everywhere, right? So nothing special here. So what I think is more interesting is this analysis that we did here. So let me help you simplify this. Again, here we have columns. Every column is a mouse tumor sample, and every row is a patient. This is from the TCGA. These are gene expressions. These are RNA-seq. So what we did here, because in TCGA we have pairs of normal tumor, first we identified the 800 genes that are changing or are more variable when you compare tumor normal. Then with this set of genes, we calculated the correlation between every single patient with every single mouse sample. This is what is here in the heat map. And based on these correlations, now cluster the patients. So we are clustering the patients based on the similarity, the correlation of that sample with the mouse models. So these patients are correlated, or are clustered based on similarity to mouse models. So here we have three main clusters of patients based on these similarities. For example, H1 includes all the normal samples here, plus a bunch of tumor samples. And the main driver of this clustering is similarity to tag 1. So if you remember, tag 1 was adenoma-like, so clusters with normal and these tumors. There is some contribution of few samples of stem and MAC, and DN a little bit, some samples, but it's weak. Then we have another cluster here, smaller, the H2. And this cluster is driven by the similarity to around half of the samples of the stem. And finally, we have the H3, and these patients are clustered basically with the similarity of stem. Okay? So now we can start seeing if there is any interesting behavior of some molecular features. For example, proliferative signature. This has been described to correlate very well when you cluster patients based on gene expression. This signature is very strong and is one of the main drivers of the first separation of patients. So this signature is highly enriched in H3, for example, while in this two cluster is not. Obviously, this correlates with the Hoshida clustering. For example, H3, we have C2, clear engineered C2. H2 is clear engineered C3, and H1 is a mixture of C1 C3. And again, grade will correlate with all these things. So H3 is also enriched in this group that is highly proliferative, while G2 and G1 are more enriched in these two groups of patients. When we look molecularly, for example, beta-catenin, now you can see that this cluster is hugely enriched of beta-catenin activating mutations. And this correlates with this set of samples, stem samples that have also beta-catenin activating mutations. So all these similarities are driving this cluster. And we can see that the different patients, or every model, what is similar to in human. So the conclusion so far from all these analyses is that the spontaneous mouse models with chronic liver damage, the TAC1 and the MAPUPA, have higher similarity to human HCC in terms of mutational processes. But mouse tumors tend to be low grade with few or known oncogenic mutations. The stem tumors are the more similar to human high-grade HCC, but the problem, or the caveat, is that it has a much higher mutational load to alkylating process. And the DN mouse model should be avoided, in our opinion, because the tumors are less similar in all analyses we have performed, are dominated by BRAF or HRAS mutations that are really found in human HCC, and moreover, they don't arise in the presence of chronic liver disease. So we don't see any point of using this model anymore. Finally, we wanted to perform an additional analysis with these data sets based on the concept we published recently about the MHC, how the MHC genotype, MC1 genotype, restricts the oncogenic mutational landscape. The concept here is that the MHC1 genotype of patients is associated with specific oncogenic mutation probability. This means that patients, based on the combination of alleles, they can present certain oncogenic mutations. And what we demonstrated is that depending on this coverage, mutations that are not easily presentable by these combination of alleles are more likely to happen than mutations that this patient can present. Obviously, this phenomenon in the patients translate in the population level. So oncogenic mutations that are highly frequent in the population will tend to be less presentable across the patients. And mutations that are less frequent tend to be more presentable across the patients. And again, this translates that recurrent oncogenic mutations tend to happen in peptides that are fully presented at the population level. So this means that for an oncogenic mutation to be highly frequently found in the population, it needs to be oncogenic, able to transform cells, but also be full across MHC1 alleles, human MHC1 alleles, otherwise you wouldn't see it. So we thought that these concepts are relevant in mouse modeling because different genetic backgrounds have different MHC1 alleles. So it can be important which background you use, and if they are different, you can get different mutational landscapes, for example. So what we did is, the first thing we did is here we have what we published in human. So we compared driver and random mutations. And as we published before, the driver mutations tend to have scores higher, means less presentation compared to the random mutation. And this is conserved across the species with the exception of pig. So here we have a lot less alleles available in the databases, but this is what we could do. So if we focus on the HCC mutations now, in human and in mouse, here we are comparing driver and passengers. Again, you can see that the drivers tend to have higher scores, meaning less presentation. So this phenomenon is conserved between. And then we did a thought experiment to ask what will happen if we use two different backgrounds. So in our mouse models, we use BAC6. So, and this is the real data, the real mutations we got from every single mouse model. And we asked what is the presentation using BAC6 of all these mutations, and what would be the presentation if we had used the BALB-C. And as you can see, BALB-C tend to present much better on average these mutations than the BAC6. Mainly this is due because BALB-C has 3 MHC1 alleles, while BAC6 have only 2 alleles. So that's expected that you will have more presentation. And of course, this translated in passenger and drivers. Both types of mutations will be better presented in BALB-C. So our conclusion is that if we had used BALB-C, most likely we would have seen a different landscape of mutations based on the MHC genotype. So that's a conclusion. So the genetic background can have profound influence in tumor induction based on a lot of things, for example, different metabolism, but also in part can be due to MHC1 difference.
It seems like there was a much bigger difference in the mice than in the people on your previous slide. Right there in the upper right. What do you make of the difference between the human?
Because here the number of mutations is hugely different. And here we have many more mutations. So this is to me the point is that we don't have many drivers here. I think we have only 27. But in here we have many more characterized. That was the main difference.
So do you think that what it's saying is that in people there's very little difference between the driver mutations and the passenger mutations? I mean, that's...
Yeah, maybe yes. Yeah. It's so slightly, is in the limit.
Yeah, it looks very similar.
So this is very strong because there are statistical problems as well. So when we use all mutations, you have much more power and all the alleles.
Right.
But when you start using only cancer types, you have less patients, less data, and it gets more tough to see difference. And in the mice, it's extreme. So we see more, but I don't think that's real, that we have much more. It's just because we have less data, and the chance is very different. That's my interpretation.
So, Joan, you were saying that the HLA1 allele impacts the mutational burden that you see in some patients. Is that correct? So, different alleles will have different mutational burdens.
Is it mutational number or the specific mutations?
Specific mutations. So the specific MHC1 combination will determine which mutations you are more likely to acquire if you develop a tumor.
But so the MHC allele determines the likelihood of the mutation being presented, right? The peptide.
Yes.
But the DNA has not. So the mutated DNA, the mutated P53, has nothing to do with being presented. What is presented is the protein. Right?
Yeah, of course.
So what links this is to neoantigens rather than actual mutations.
Yeah. We are talking about neoantigens. But anyhow, the mutations are at the end translated in a mutated peptide
Yes.
that could be presented or not. Yeah.
maybe I'm confusing something, but don't, like each mouse strain has specific subtypes of allele,
Yes.
or is it genotype, and maybe like A to D or A to C?
Yes.
So wouldn't, to look specifically into that, wouldn't it be more informative if you would have used the same background strain, like C27, like 6, but then have them on different haplotypes to really say it's MHC dependent, or
Well,
like,
so
because
the,
the BOP-C and the C27, like 6, they have many different genetic traits, right? And for example, for fibrosis,
Yes.
for the immune system, they have different amounts of inflammatory
Yeah.
cells. If you isolate, they have BOP-C have many more stemning cells. For example, you get 2 to 5 times as many cells from one liver than from the C27, like 6, for example. So could there be also other things that?
Yeah, of course. I think there will be a lot of factors.
Mhm.
We were just suggesting that there could be an additional one, that is the NHC1 genotype, that is associated with different backgrounds.
Yeah.
That could be another factor to consider
Mhm.
when you're using different backgrounds. This is a thought experiment. So the real experiment would be to use black 6 and genetically modify the NHC1 allele, having all the remaining things exactly the same, and then induce the tumor and see how the mutational landscape changes. But it'd be great, but it's a long time. Okay. So, in our goal to improving HCC mouse modeling, at least I think we can say that we shouldn't be using the DN, right? There is no chronic damage. The dominating mutations are never found in human HCC. The mutational process is never found in human HCC. Gene expression profile is the less similar. So I think this model is very popular. We've been using it for years, but I think we should stop it, sincerely. On the other hand, we have a STAM model that is targeting similar genes. We have similar mutations. The gene expression profile is quite similar. Histologically, is also similar. The reason the problem is that it has a huge amount of mutations. And that can be a problem depending on what you're trying to study. If you're trying to study immune response, well, we have such amount of neoantigens that, so it depends on what can be useful or not. For now, this is the best model of the 4 that we have. And then TAG1 and MAPIPA can be useful if you're interested in analyzing how the mutations are acquired in the path towards HCC, because the way these mice acquire mutations is very similar to a human HCC. So in order to keep improving or going further in the mouse modeling, well, I was in favor at the beginning on trying to translate very faithfully and specifically the genetic alterations that we now know that exist in human HCC. But I'm starting to change my mind. So the reason is because, for example, here we have the top 10 mutated genes in human. I think this is from TCGA only. So here we only have the patients that at least have one of these genes mutated. You can appreciate the complexity here. So if we want to translate this complexity, I think it will be difficult. So we have, for example, patients that p53 beta-catenin, p53 beta-catenin, any other of the other 8 genes, p53 but not beta-catenin, and then all the other genes, beta-catenin but not p53, any other genes, and so on. And
what
then
about telomerase? I'm surprised. Isn't telomerase one of the most frequently mutated genes?
well, probably here we were considering the coding sequence only,
so yeah.
because usually a mutation is in the promoter.
But don't you think for mechanistic studies it's kind of important to include the non-coding?
Yes. So we never found anything, especially in the telomerase in the mouse models, but this is a fundamental difference between mouse and human.
Yeah.
So mouse, almost all cells have active telomerase. This is why it's easy to immortalize mouse cells. And humans is completely different. So that's a fundamental difference, so that we need to know about. And here, we never see overexpression of telomerase in the mouse models because it's already expressed. So that's a fundamental difference. but we cannot do, I think, anything.
Unless you worked in a telomerase knockout mouse.
Yeah, but then you need to somehow be able to put it back, overexpressing it, if you want to try to model overexpression of telomerase. This is another complicating factor.
When you show this complexity, do you think that it could be the case that a given patient, one or two mutations are the key drivers of tumor progression in that particular patient, and the rest, despite being mutated, potentially damaging, they're not so relevant? So to what extent do we need to precisely recapitulate the whole set of mutations that a patient has, just focus on the 2 or 3 that are, the key drivers?
Yeah, I totally agree. And this is what I was trying to say here, and how precise we need to be. Because if you want to be really precise, you should start translating exactly the alterations you have. Right? Splice sites, nonsense, deletions, activating mutations, only one. There are several activating mutations. You need to mimic exactly the same alteration in every case, so that the complexity can just explode. And I think going in your direction, so if you look, for example, this is a figure from the TCGA paper. So the famous I clusters, right? So they have only 3 I clusters based on copy number, methylation, expression, and microRNAs. But to me, and of course, you have this segregation of molecular features. But to me, what is surprising is that if you look, you don't see much subclustering. Right? You have these 3 big clusters, but I don't see many subclustering here. Looks more or less the same. And to me, the concept is going in that direction, that maybe a few molecular alterations have phenotypic dominance, and the other ones are just complementary. They're there, they will do something, but they're not defining the phenotype very clearly.
So how do you distinguish those 2 types of mutations?
Well, yeah, well, I think it's relatively, the obvious ones are the ones that are segregating clearly with the clustering, right? So the alterations that segregate clearly with the phenotype should be the ones that are potentially the drivers of the phenotype. So I would focus on these first, and they are no surprise. So, beta-catenin, CDKN2A, P53, and so on. So no surprise here. But I think what this is suggesting is that it might be that we don't need to be so precise in trying to regulate every single alteration you find in a patient, because maybe it's not so meaningful. But this is just an open question, because it could happen that you don't see much subclustering for the same reason you have this huge complexity. If instead of hundreds of patients, you have 50,000 patients, maybe now these groups will be much bigger. And in here, you will be maybe you start seeing subclustering, I don't know. So that's another possibility. But to me, it's very important that we address this question so we know how we need to model. We really need to be very precise, or we can just model few alterations, and with this we are having reasonable mouse models of humanists. So the other observation relevant for modeling the disease is the risk factors and etiology. So if we look how they associate with the different genes or altered genes, there's no much association, right, between HBV, HCV, or cirrhosis, depending on which gene you're altering. And if that's true, this is another important thing. So if the etiology underlying chronic liver disease has little molecular impact on HCC features, then you don't need to be very precise in how you are mimicking the chronic liver disease. You don't need to have HBV chronic infection in a mouse to really be faithful to the disease. And that could be very important for modeling, because it's not so important how you produce the damage, is that you have the chronic liver disease. So I think answering these 2 questions would help a lot how we need to approach the mouse modeling of this system.
Well, certainly the second comment makes sense, because there are no mutational patterns that are unique to specific etiologies. They cross etiologies. So it seems like the common feature is more chronic hepatocyte injury and all the changes that elicits, including fibrosis.
But I think this is important because we have so many models of coronary liver injury. You can do it in so many ways that just to have a general way to do it that have the same impact that any other one, that could help make everything easier. Nowadays, you need like 5 models to be able to make any conclusion, right?
Or they get published itself.
Yeah. Okay, so this is the end of the first part, and now I'm starting quickly. We are on time. The second part. That is about the role of SOX9 in cholangiocarcinoma development. So I pointed out before, in the wild-type liver, SOX9 is expressed in the bile duct cells and in these hybrid hepatocytes. Expression here is much higher. So we decided to produce a liver-specific SOX9 deletion, trying to understand what could be the role of SOX9 in the behavior of these cells and in liver cancer in general. So we generated this crossing, our increased SOX9 for flux, and this is deleting SOX9 intrahepatically in the bile duct cell as well. So here you have the CK19 positive cells. There is no signal of SOX9 anymore. So we are deleting in hepatocytes and in bile duct cells. So these mice look normal, more or less normal. So it's not uncommon to see some cysts, formation of cysts. The histology look pretty normal, but sometimes there are some problems. And when these mice age, it's more common to see some cysts and more problems and infiltration in the portal tracts. But in general, they are normal. So the first thing we did is try to see what could be the phenotype of oval cell response, since we don't have SOX9 in the bile ducts anymore. So the wild type, as expected, you can see the CK19 positive cells expanding, the typical oval cell response, and in the SOX9 deletion, we still have the bile duct cells, bile ducts, but there's no expansion or proliferation at all. It's completely blocked. This is an independent model, the CTE diet, another model of oval cell response, CK19 expanding cells. SOX9 deletion, we still have clearly the bile ducts, but there's no expansion. So this clearly indicates that SOX9 is very important, not to maintain the bile duct cells because they are still there, but when they need to proliferate or expand, it's required. So the other thing we wanted to try or test is it's well known that hepatocytes can transdifferentiate to ductal cells in cholestatic injury. And SOX9 is a very clear marker. When the hepatocytes transdifferentiate, they start expressing SOX9. So we were wondering if SOX9 could be important in this process. So it has been reported that notch signaling is very important in this process, and when you express notch intracellular domain in hepatocytes, these hepatocytes transdifferentiate. So we generated this transposon, that is a notch intracellular domain together with GFP, and we injected this transposon in SOX9 flux. This is a wild-type mice. And as expected, the hepatocytes that are taking this transposon change completely morphology. They start expressing high levels of SOX9, and they become CK19, so ductal phenotype. So that was working. And when we did the same experiment now with the deletion of SOX9, you can see that these hepatocytes are taking GFP. They retain the morphology. They don't express ductal marker or cell-pontin, and they retain the expression of HNF4 alpha. So the transdifferentiation process in the absence of SOX9 is clearly hampered. So discussing with Hayato Nakagawa, it became clear that since SOX9 is important for bile duct proliferation and hepatocyte transdifferentiation to bile duct, it was obvious, right, that maybe you have a role also in the cholangiocarcinoma. So maybe SOX9 is required for the formation of cholangiocarcinoma. So as you know, the classification of cholangiocarcinoma is anatomical. So we have intrahepatic cholangiocarcinoma, perihilar, and distal cholangiocarcinoma. And the incidence is not very well established. What I understand is that the extrahepatic disease is more common than intrahepatic. And another feature that is very clear now is that the intrahepatic disease is increasing. The reason for this is unclear. These are the numbers. Etiology in most cases arise de novo, but it's very clear as well that cholangiocarcinoma is also associated with chronic liver disease, primary sclerosing cholangitis, and in Southeast Asia, because of these parasitic worms that infect the hepatobiliary system, the incidence of cholangiocarcinoma is really, really high. So in collaboration with Hayato and Tsuneke Noe, University of Tokyo, they developed this mouse model of cholangiocarcinoma that is the albumin Cre-P10 KRAS. So these mice developed crazy, this is huge liver at 8 weeks old, and all they die by this age, without exception. And histology reveals that this is cholangiocarcinoma. I'm not an expert in histology, so I wouldn't know how to classify this. It's supposed to, and it goes really, really fast. So before 3 weeks, everything looks more or less normal, but in the transition to 5 weeks to 7 weeks, there's an explosion, and these mice basically all cholangiocarcinoma everywhere, and they just die. So importantly, in their report, they wondered if, since they are using albumin Cre, this could be deleting in both compartments, bile duct cells and hepatocytes. And it's unclear this cholangiocarcinoma from where it's arising. So this is why they use the albumin Cre RT2, that with the addition of tamoxifen is only targeting hepatocytes. So now, in these conditions, they are deleting P10 and KRAS only in hepatocytes. And after a long period of time, the tumors they got were HCC. There was no cholangiocarcinoma. On the contrary, when they did similarly with the CK19 PRT2, at 6 weeks, very quickly, they developed cholangiocarcinoma in the distal part. But these mice all died very quickly because they not only generate cholangiocarcinoma, but also lung tumors, because CK19 is expressed in a lot of different tissues, so these mice were really full of tumors and died. So this is also suggested that in this model, the origin is bile duct cells. It's a relatively bona fide, I would say relatively now, after talking with Daniela, bona fide genetic model, because KRAS is usually found in cholangiocarcinoma, P10 not so much. It's SOX9 positive, it's highly aggressive and fast, and it generates intracholangiocarcinoma, intrahepatic cholangiocarcinoma, and perihilar. Both types, because it looks like albumin Cre is not only targeting the most external part of the bile ducts, small bile ducts, but a little bit in the perihilar region. So what we did next is to use this mouse model combined with our SOX9 flux to see if there was any effect. And there was a clearly very significant increase in survival of these mice. And you can appreciate the difference of 5 weeks, the liver size and the amount of tumors, the histology. These mice were like full of cholangio. And with the deletion of 9, you start seeing appearance here, but it was still quite normal at 5 weeks. But these animals, regardless of this difference, they finally died. And when we analyzed these terminal mice, we found something really interesting, is that in the small bile ducts in the periphery, they were normal. They were normal bile ducts, they were not transformed, there were no cholangiocarcinoma here. But it was coming from the perihilar region. So our interpretation is that maybe intrahepatic cholangiocarcinoma is blocked, but the perihilar not, and this is what at the end is killing the mice. So that would be the situation, right? The SOX9 is important only in intrahepatic, but not in the perihilar. So the conclusion so far is that in this mouse model, we have origin in ductal cells in both intrahepatic and perihilar, and that one is SOX9 dependent, potentially, and another, the perihilar, is SOX9 independent. So what we are trying to do now to complete this project is to have an additional mouse model in which now we have cholangiocarcinoma originating from the hepatocyte transdifferentiation to see the complete picture of SOX9 relevance in both situations. And the other thing we are trying to do is we have established these organoids from CK19CRT2, and we are trying to isolate them from different regions, intrahepatic and perihilar or distal. And the idea is that inducibly now, because unless there is tamoxifen, these organoids are wild type. So now inducibly we can induce the formation of cholangiocarcinoma in the presence or absence of SOX9 and try to understand what's the difference between perihilar and intrahepatic, and what's the different role of SOX9 in intrahepatic or perihilar using RNA-seq transplantation. So this is the end of the talk. These are obviously still ongoing. For the first part, this was in collaboration with Michael Karin, Hannah Carter, and Michelle. That was the first author of the paper. The people in my lab, Lauren, that is driving the cholangiocarcinoma project, and Nicola, the HCC project, and the collaborators at the University of Tokyo, especially Hayato and Suneo. And this is it. If you have any questions,
Questions? Sunny. Okay.
In your SOX9 knockouts where you prevented the ductular reaction, did these mice go on to produce tumors?
We didn't follow up longer than that. So we just did the typical 2-3 weeks treatment, and that's it. We didn't go further.
Sorry, do any of the mouse models have the sort of male dominance that we see in human HCC?
The DN definitely, the stem completely. So if you do males, I think it's 100% penetrance, but females is difficult to get. And I think there will be a relationship between the carcinogens. I think the carcinogens are more effective in males than females.
And
then
so
the type 1, I think there is no difference.
in the STEM guys, did you sequence the non-tumors? So you say you didn't get tumors in the females. I'm just curious if you have a similar background incidence or of mutations. How do you rationalize? It's very interesting. How do you understand the difference that you're seeing between the males and females in that model?
So what I would do is to do a parallel situation with the DN, because in the DN we know much more. And I think the models are relatively similar. We have a carcinogen, if you then the stem, they are diabetic, and then you have a diet, you have chronic damage in the DN. But it's very clear that in the end you have this male-female difference, and mainly is due to the difference in inflammation and the regenerative response after the administration of the carcinogen. So you have more damage associated with the initial damage and the compensatory proliferation you have after that in the males and in females. And probably this is the reason why you have more mutagenesis in the males.
So you have more inflammation in the males?
Yes. Yes.
In both models?
I don't know in the stem, but I'm just parallelizing from what we know from the end. So my hypothesis would be that the stem works somehow similar. When you inject STZ, males, you will see more damage, more compensated proliferation at the beginning. So then you have more mutagenesis compared to females. That would be my hypothesis.
Thanks. Interesting to know.
Yeah.
I'm really interested in the SOX9 model where you get the extrahepatic but not the intrahepatic. So you say that extrahepatic is clearly involved in the large bile duct?
Yeah, because we see that in the terminal animals, what is more, because it's confusing to me. So the small bile ducts, you clearly see that there is nothing in the small bile ducts, but the tumors are located in the big bile ducts.
But it's invasion because they're quite big, right?
Yeah. Yeah, when they are terminal, it's just over the place. But you can still see that in the small islets, they are still normal. But the point here is that we don't know the owning tree until where is active in the ductal tree, until where deep is active. So what we are trying to do is to overcome the owning tree and doing with another method to really pinpoint the anatomical locations and be sure that that's correct. Because we cannot rely very much with owning tree because it's unclear.
Joan, last question?
Yes, I think Joan, you already mentioned, but I think it's clear that cholangiocytes within the liver, like the intrahepatic and very high other probably from the large bile ducts, are very clearly different in their behavior. And then even you already also talked about the gallbladder. So I think you should really go ahead and then do some additional analyses from trying to tease it out or laser microdissection and highly interesting because it's such different type. And then maybe the other thing with the SOX9, I found it interesting that with the albumin-free SOX9 mice, CDE or DBC diet, you do get CK19 positive cells, but they're not as expanded. But they seem like the blobs are so they are more. I think it would be interesting to find out maybe how SOX9 is impacting these cells. They are still clearly there.
Yeah. No, I think that's very interesting because SOX9 is not like the bile duct cells are still there and have identity, but their behavior, their processes are completely changed. And I think what I'm trying to understand is what is changing, why this is important. Because when you remove nodes, you don't have bile ducts. The body of ducts are gone. So SOX9, you still have the bile duct cells, but they are somehow compromised in the way in their functionality.
And one last observation, maybe you could work with the pathologist because I thought like very high the tumor, and then the area which was not affected, it seems you had a lot of vessels, and this can also indicate that there's kind of atrophy because the liver lobular, they're getting smaller, and this is why relative for your field of vision you have more vessels. So
yeah, definitely I will send samples to Yeah, of course, sure.
Okay, thank you very much.
Thank you.